last topics which we discussed was evolution, and the one we haven't started was about viruses, because viruses is a part of life, though we can kind of start arguing if they're alive or not alive. And the last discussion I had, it was a 5-year-old girl with a professional biologist, and they have different opinions, and of course they're equally naive talking about that, yeah, because it's just a question of language, yeah. But definitely the crucial part of life. For each bacterium there are probably 10 viruses, and that for each kind of cell, whatever in the world, there is dozens of viruses, and they influence tremendously development and evolution. And it's the major, at least as far as unicellular organisms are concerned, we are one of the major factors in evolution. However, there were others. And so the remarkable mathematical aspect of viruses, which is mathematics, which is behind them, there are many, of course, many mathematical aspects of their structure, but the one which was discovered by Crick and Watson, in my impression, it was actually by earlier paper by Watson prior to their work on DNA. And so there is this thing about viruses that they solve the following quite remarkable mathematical problem, which mathematician has never approached, and still we have difficulty of formulating it. And it is as follows. So the fact which was observed by Watson, I think in the 50s or in the late 40s, was that at least some viruses have the dodecahedron icosahedron symmetry, yeah, which you know well. So they have shape, not exactly maybe icosahedron, but they have this symmetry, right, which is a quite remarkable group, as you know. It's the same as permutation group mod center. And of course, at the time, it was not that you look at them and you see them. Yeah, you know that you look at what you see. You know, it's not was like that, because they are invisible. And at that time, there were no electronic microscope photographs of them. Now we do have them, and I think I've shown them before. But it was by indirect indication of their incomplete X-ray kind of pictures. And as you know, X-ray crystallography, when you crystallize them, they were known to be, some of them could be crystallized. And what you do, you have kind of a absolute value of the Fourier transform. So you don't even have Fourier transform. It's just part of the, certainly the very imperfect pictures. And from them, however, Watson made this guess, and his justification was, so why this form is preferable? Of course, it's not the only kind of symmetry. There are two typical symmetry. One icosahedral, and another is just helical symmetry. The tobacco, on tobacco leaves virus, it had this kind of symmetry. And this is, of course, the most primitive symmetry, because it's just one motion, one rotation, but you continue it many times. And so it's again, it's a process which actually starts and repeats, forced to be processed this time, many times. And then there is icosahedral, which is kind of more elegant and more complicated mathematical symmetry. And so why it is chosen by viruses, not by bacteria? And the reason suggested by Watson is, again, just looks completely correct, and was then was systematically viruses, at least small viruses, I'm not certain so much about larger viruses, have the structure. And the reason is that viruses are supposed to be small, and some of them have 3, 4, 5 genes. Well, I'm exaggerating, maybe, but less than 10 genes, some of them. So they don't have much information they carry, and they have to contain this information in proteins encoded in this DNA. So there are some number of genes, and it must be realized that each gene, all this DNA sequence, it may be almost 10 times bigger than the protein it encodes. Right? It's very difficult. We have a library, so don't people run away. Yeah, okay. So I'll finish probably earlier to keep you. So that's a remarkable kind of, it's a library where books describing a building bigger than the building. And then you have to do that, and so you have to minimize the amount of information and maximize kind of the size of this enclosing surface of them, right? So you build this surface, follow some instruction, and you have to make this ratio, probably the ratio, maybe not the right relation, but say a ratio between the amount of information and what you build to be minimal. And therefore, it's very advantage. But what you can do, although each particular gene as an encoded DNA is much bigger than corresponding protein, but you can produce many proteins. Actually, not you, but how virus doesn't produce its own protein, right? It sits inside of a bacterium and tells bacterium do it. And bacteria, or your cell, yeah, not necessarily bacteria, it also applies to your organism. A virus comes into you and tells you, replicate me and produce many copies of myself and many copies of my proteins. And you do that. You say yes, yes, yes. You don't argue. Right? Virus is much smarter than we are. And then we shall see, we're now approaching, our technology approaching the intelligence to viruses. It is next stage of the evolution on Earth, will be when people, humanity, will function as efficient as viruses do. And then, so you may have many copies of the same protein. And so if you have just a protein which has such a shape that you can assemble out of them, for example, icosahedron, so you produce the same copy many times. And many times it costs nothing to the virus, it costs something to the cell, which does it, but not to the virus. So many copies are produced, and then, remarkably, and this is a physical and non-trivial physical problem, under certain properties of this protein, there is self-assembly. When they're floating many molecules, say identical, for simplicity, which happens for very small viruses. For bigger viruses, you have several copies, several kinds of these molecules to fit perfectly, and then they flow, and then with high probability, they make this shape sitting with the DNA sitting inside. I don't know how much it's understood. Yeah, it is a almost purely physical process, I guess. Nobody guides them, right? Because virus has small virus has no gene to make them do that, and cell also doesn't exactly adapt it for that. But just floating and with certain density, this happens with high probability. Probably many of them not unstable, but viruses don't care. Some do, and so they go out with bacteria. And this is an interesting mathematical question, how it can be done. We can formalize the individual steps, and here, as in the case of protein folding, there is this particular thing which happens as is kind of basic physics thing, flow with some probability molecule touch each other. There is probability they stick together, and so some self-assembly takes place. So it's a random process, but it is controlled random process. You create parameters, and so and this is of course reminiscent of the classical isoperimetric problem, when you have to contain maximal volume in the minimal with minimal area. Okay, but now, because there are not too many of you, I would rather entertain you by pictures to what happens next. I just said it about viruses and about evolution. I wouldn't tell much, but just show you the pictures and of what comes next. And so my first lecture was about structure of molecular world and life from this molecular perspective. And here it will be something else. And this is the same, but from the perspective of our planet and humanity, which became dominant, at least as far as multicellular organism, or at least mammalian organism, we became dominant. And so we influence very much what happens here. And first we look at the history of Earth to see how it was happening. Right, this is what was happening, and we see that the major phenomenon which took place was photosynthesis. And this is before apparently the Earth had no oxygen, and now life is mostly dependent on oxygen. And at the time of the origin, oxygen was poisonous, and it was produced by cyanobacteria, and then they were dying as a result, and there were some pulses there, but it's not fully understood. Yeah, so you can fantasize about details of what was happening, but there is a reason to believe that the oxygen which we have is a result of early life, of the first billion years of the life. And here is how it was happening, and then in some moment, and about 2 billion years ago, eukaryotes came up, and then pretty soon afterwards, everybody else. And as far and from certain perspective, we haven't changed much after first eukaryote emerged, yeah, compared to what was before that. However, it was not that smooth as it may look. And again, the major factor was there were at least two essential processes which were involved, and one of them, as you see, the photosynthesis. And because of the way carbon was fixed and brought to life, and without that, life now will be impossible. Almost all life would disappear, but original life were not depending on the light, yeah, for all we know. Right? At least it had to imagine how from nowhere you could reproduce, because, as I shall say a few words later, this photosynthesis is a very complicated process, and it couldn't just come out of nowhere. And it came in several stages after already we had rather developed cell, and then there were again, this problem is just doesn't want to. Okay, this is a picture. That this is a graphic shows how many animals were dying in certain periods, or not animals, just all kind of live organisms. They were in the observable time. There were 5 major extinctions, and the last was dinosaurs, which were apparently destroyed by the impact of meteorite on Earth, and conceivably became coming to the 6th created by humanity, which is similar to what's happening with cyanobacteria, right? We produce something which is poisonous for the environment and, first of all, for ourselves. But this is, as you look at that, in this oscillation picture, you kind of understand, because there was controversy in all the time between people like Cuvier, who was in the beginning of the 19th century suggested that evolution was following by a sequence of catastrophic events, and then everything was starting again. And some people like Lamarck and then Darwin insisted everything was nice and smooth. And just looking at the picture, of course, it depends on the scale. Along each line, kind of smooth, yeah. But on the large scale, it's certainly very oscillatory. And the major event, most relevant for us, was a Permian extinction about 2,050,000 years ago, without which we would never be here. You know, it was a major fact that determines, which brought up all organisms like ourselves, a sufficiently developed system, so active system of respiration. We are not as good in respiration as birds are, but certainly much better than animals which were before that. And because the life there was very tough, there was not enough oxygen, there were many problems, it's not fully understood what was happening. But the major extinction, this is, I think, is a Permian extinction. Yeah. So it is, you see it, yeah, it's in there. So this was happening there. And so this is very instructive to think about that, yeah, that anything which you say about biology, unless you know it, will be wrong. Yeah. Things happening There are so many factors. This gave you see, what was before there were this, of course, extinction due to production of oxygen, and then there were periods when the ice, also prior to that, when the Earth was just all snowball, which we know less, but this is a kind of rather contemporary events, which happened after Cambrian explosion, when already there was very well developed life on Earth, which we know a little bit about. Now, so there are two topics I have to briefly to cover, and here they are. You can see them. Now we go to that, and now this thing does move. Okay. Okay, so this is kind of a little schedule how happened, how we can look from mathematical perspective on that. So, and from mathematical point of view, of course, the major problem is language problem, I believe, and so maybe it will be solved, and I'm using it will be solved by specially created mathematician who would undergo at some point say, a few years from now, you develop better gene manipulation than we have. For some reason, what we have, at least as I understand it, it's still the best system we have of gene manipulation, is CRISPR, is still not good enough to apply to human because it has too high a level of errors. And but there are kind of, I think, good perspective will be improved, and just we shall see that all kind of before everything was in medicine, or unless the last maybe 30 or 40 years, medicine on biology, if you're more or less on the level of Pasteur or Jenkins, when people were just making something without understanding the structure of the object during quiz. And now, all the last events, on the contrary, were result of a much higher, absolutely different level of understanding the cell, and some manipulation with organism needed for experiments and for production. And some results which we've seen, as I said before, this was new vaccine for COVID-19. This is a manifestation of a new level of biology, completely having kind of incomparably different from all previous what vaccination was. It's as difficult as bacteria from eukaryotes. There was completely new level, and this is will of course will change the course of human history pretty soon, включая mathematics, because we will be able to breed either individuals or human brains. Maybe, of course, artificial intelligence will go ahead. We don't know. But so far, biology does much better. So, but whether we solve it or not depends, of course, how the Anthropocene develops. So now we see what happens there. So, what is a kind of major thing we have to know? Again, indeed, it is somehow unpredictably how things are being regulated. So it doesn't move as they push it like that. Okay. So now we have lots of pictures. So besides this photosynthesis, which was fixation of carbon, in this kind of the major thing which makes us there was second process involved. And this was fixation also by bacteria, is fixation of nitrogen. Because we have a lot of nitrogen atmosphere, and nitrogen is essential part of your body. In a second I show you the picture. And not only your body, but everything, because proteins contain nitrogen. Because before that, when you have only carbon, oxygen and hydrogen, you can produce lipids, and you can produce sugars of all kinds, but they're not enough to make life. You need for some reason, we don't know why, but we need two other elements, and they are nitrogen and phosphorus. Of course, many other things are involved, but these are chemically the major kind of ingredients. But we concentrate on nitrogen, because we have nitrogen atmosphere. Unlike phosphorus, we do have it in soil in a slow process, but say plants can slowly absorb it, but not about nitrogen. Nitrogen can be fixed. Atmospheric hydrogen is not useless, and there are two-way fixation, whereby due to the lightning, they create some oxide of nitrogen which doesn't go to the soil, and then there are bacteria which do that, and this happened again in the very early stage of life before photosynthesis, right? Because all should have happened before photosynthesis. There were no oxygen atmosphere. Photosynthesis too complicated. Probably this nitrogenase, these enzymes appeared before photosynthesis. But this what we show is production of artificial production of nitrogen, of ammonia, which was invented, as I indicated on the previous slide. It was July 2nd, 1909. So July 2nd, 1909, we moved to Anthropocene. It's very remarkable. We have specific date. We know exactly when it happened, right? So we wouldn't be here if not for that. It was a specific event, and then 4 years later, production of ammonia, industrial production began. And from that moment, you see what happened to the population. We see how this thing correlate. Production of ammonia and human population, because it was a major bottleneck which is limiting food production. And now we produce as much of ammonia as all bacteria on Earth. And most of them, of course, go to, not for us, they go to, in the seas, they go to the forests, and all they depend on this bacteria. And then they say a couple of words how it works. It's an immensely complicated process. It's not fully understood, and we cannot imitate it. And we have artificial ammonia production. It's a very kind of messy and noisy and dirty process, which is very much polluting, requires much energy, it's very unpleasant. However, 8+ billion people who live there depends on that. So if you cut it, 2/3 of people will starve, yeah? Almost instantaneously. They depend on this mechanism of Haber-Bosch process. Right, so it was in this historical moment. Actually, people don't realize it was the most important event in the human history probably after the creation of agriculture, about 10,000 years ago, approximately. But then there was no specific moment. And this was a specific moment, and there was Haber who made this experiment, and Bosch, who was present, realized it can be done industrially. Because the point is that it was clear, of course, to chemists already at that time, that you only can do it under high pressure and temperature. But you needed also a catalyzer. And the catalyzing, of course, tricky thing what it is, yeah, but anyway. And originally it was, I think, bismuth or something used for catalyzer, and then now we are using just just iron. But anyway, and it took 4 years, yeah, and then commercial production began. So I think you just have to emphasize, this is a key moment in human history, at least for the last 10,000 years. And it's not some wars, whatever. And this changed the face of the Earth. Everything else was small in comparison. Electricity, nuclear energy, I mean, it wouldn't affect much. That it was more or less the same. Yeah. It was minor. It contributed to some other aspects of our life, but not to fact that we became so many of us. And how many, so we shall see. I'll show you the pictures. So I just want to, and it's never in any calendar, it's not being, I don't know if you have celebrated or on the contrary, maybe it's a bad thing which happened here. This, of course, the major, so the next major event, of course, will be solution of Lang's problem. Then because maybe we'll celebrate it. Now we have to speak about, this is a useful picture. So how different elements distribute in your body, just to have the idea. But mainly weight-wise, of course, oxygen, because it's part of the water, because about 60 to 70% we use water, we were made of water. there is a lot of hydrogen there, but hydrogen very light. Atom-wise, of course, we have lots of hydrogen, but weight-wise, the major fact that you see this table shows very well, and how it goes. And nitrogen comes just after oxygen, carbon, and hydrogen, and then nitrogen comes. Phosphorus is not so visible. Phosphorus slightly less, but phosphorus is kind of, well, it's another story with phosphorus. Phosphorus is a rather common element on Earth. It's, I think, about 12 or 13th most common element on the Earth's kind of not surface, but the crust. And but it's still we don't have excess of that. There's one big problem with phosphorus, that is being resources of this systematically being depleted, unlike what happens with say, nitrogen. It goes usually back to atmosphere, and we lose phosphorus all the time because it's in some minerals and then it's used and then just become unusable. So this is the reason. But here is interestingly, so what we have, and you see the last, we have, so we have a few words, yeah. Probably you know the function of all of them, yeah, in the body. And selenium, for example, come in certain proteins. It's in one of the 21st amino acid. There are 20 basic amino acids, and one of them has some variation where sulfur replaced by selenium. So I don't know exactly what it does in your body, but it is there. And the last is molybdenum. And molybdenum is not that, it's as a rare, it's a quite a rare metal, but it involved in certain, into certain proteins. I gave their name, so if you're interested, you can look at the internet. But what essential, that it is involved in most common nitrogenase, the protein, if I'm not certain of saying the word correctly, the protein which is involved in production, in fixation of nitrogen. So one is photosynthesis, and the two proteins involved are, we discussed one of them is chlorophyll, which you know, and then this nitrogenase, there are kind of 3-4 enzymes which determines production of a substance, our structure. Of course, there are other proteins which makes it all this synthesizers of your DNA involved in production of proteins in general, but this is specific enzymes which take stuff from environment and put into our body. So they make connection with us, with the chemical environment of the Earth. And there are not so many of them. And well, it was worthwhile think about them, but to see how they work, first we have to look at this table. Now we want to see that. And so what is there? First I don't know, myself I kind of always forget who is who, and so I made this table of basic units of energy involved. So all depends on chemistry, it's molecular process, and they break and join molecules, and the energy must be conserved, and so, you know, some process possible, there must be conservation of energy. And so if we see it's not conserved, it means there is some source of energy. And so on this line, everything made on the scale of kT. So in biology, in our world, the major element of the energy is a average energy of the moving molecules. You know, all of them have the same squared, and it's the same, and the average is the same energy, which is remarkable. I think it just probably was understood by Maxwell and by Boltzmann, that they exchange the way they exchange, and they hit each other. Heavy molecules move slower, fast move faster. So on the average squared of the, so that the energy is more or less the same. And energy is squared velocity times their mass, and they're more or less the same. So it's the same unit. I keep forgetting. Yeah, I think the water molecule has the speed порядка 700 or something meters per second. They move pretty fast, but because they move in all directions, the momentum, overall momentum, zero, so you don't move anywhere. But this is the heat energy. And then on this scale, we can see different properties you have to know. And I spoke about ATP. ATP is the major substance, molecule, which contains phosphorus. Without it, it doesn't work. And it is used in all chemical reaction in the cell. It carries energy. If you need to go up energy-wise, you need ATP, you have to go up. And very similar, there is this NADH, I don't know how you pronounce it. It's also some molecule which may add or lose some piece of this phosphate group, and so carry some extra energy. And this involves basic processes of burning fuel in your cells. And here is the energy of basic bonds, and give you an idea how it is hard or easy to make some chemical reaction. So just, this is one of the points if you want to have any realistic understanding of elementary biology, you have to remember roughly this kind of scales. Yeah? Without them, everything is meaningless. Of course, biology, sometimes you can have this kind of on the back of their mind, because there are no consequences of that, but on the beginning level you have to know that. And biologists usually are uncareful about that, and sometimes even in textbooks, they of course don't confuse the numbers. Numbers are copies, but interpretation of them, you must be somewhat careful. So, here, unfortunately, I have some mixture. I could never manage to put pictures in the right places. Yeah, so sometimes my pictures may not exactly correspond to the text. And now we come to photosynthesis, which is one of the major things which happen on Earth. The moment it stops, so life stops, at least as we know it. Because see, originally life, so believed, originated in the hot spring, which still there are in the ocean, or something like that. It was, the input was chemical energy, which is much easier to use. But here in photosynthesis, we use energy of the sun. And it's absolutely incredible how it can be, because you have to break very kind of, I must admit I don't understand it. Yeah, I just try to look at this literature a little bit, and I say in a second what I don't understand. Maybe somebody here understands it. So it seems to me impossible. So this photosynthesis, and what is kind of basically simple scheme of reaction. So I have water and carbon dioxide, and then it transforms to something. But you see this CO2, H2O, they have huge linking energy. This I written kilojoules per mole, because they in the table they're in kilocalories, which is 4 times less. I took this from the internet, so I didn't change them, because I cannot make these nice pictures. Some people use, physicists prefer joules, of course, and chemists kilocalories. Calorie, you know what it is. Calorie, how much heat you need to warm certain amount of water, right? And I think it's 1 gram of 1 liter of water by 1 grade will be 1 calorie, not kilocalories, but I think 1 calorie. Also, I always forget where this 1,000 comes. And joules is an energy of 1 kilogram moving with the speed 1 meter per second. Kinetic energy of that. But this, of course, another thing which, of course, you have to do, and I partly done it, but not fully make this exercise, to compare all these different levels of energy. Simple, of course, we understand how much you need to warm the water, and another good kind of energy unit is how much you eat. Yeah? So on the average, we eat something about 2,000 kilocalories per day. Yeah, that's, and that's sometimes interesting compare with that, but this will come on the next level. But this we divide, of course, energy per molecule. So we divide this on the number of molecules, and mole, or Avogadro number, is a number of, in some standard amount of matter, say in 12 grams of carbon, how many atoms? And this is some big number, yes, like 10 to the power 23 with something like that. Always confused with the big number. But it was relevant for us, the ratio of these numbers, the absolute values, so of course have no meaning. So glucose, yeah, it's one thing which is involved there, and here is That is CH2O. This is a formaldehyde. It is a, but they're all kind of chemicals containing water and carbon atom in certain copies. So glucose, when we do it 6 times, but this glucose has this shape, and here is a remarkable thing. There are 2 molecules like that, of this roughly this shape. One, and the one obtained by mirror symmetry. And this was discovered by Pasteur, that organic molecule coming from life, they all have specific, choose one of those 2 symmetries. Life is not symmetric under kind of mirror symmetry. And nobody understands why, by the way. Yeah? But that's the fact. This is so-called right glucose, I think, because there are right and left. Of course, it's pure terminology. This shape is right because, you know, there are 4. The valency of carbon is 4, so there are 4 connections, 4 links it makes with other molecules, and therefore it has kind of prefers frame, and each of them has a name, and therefore there is some orientation, and may be oriented in different ways. So, and then there are these 2. So maybe I show you the process, the picture. No. I don't understand how it jumps. Okay. This is how photosynthesis works roughly. Again, first, of course, it's not fully understood. Myself, of course, I understand almost nothing. But this is a schematic picture which everybody can see how it works. And so what is this P means chlorophyll? There are 2 forms of the chlorophyll. And it first absorbs light, and then it translates it, and there is what is called a resonance energy transfer. Probably you know this, but he's sleeping, what it is. It is some kind of a quantum mechanical process. It's not kind of simple, and again, I don't know what it is. But so what is strange here, because for the photon, energy of the photon is about, of a green photon, about 200 kilojoules per mole. And so, but it must break bonds which 10 times stronger. Or at least individual bonds, of course, even individual bonds are stronger. And this, of course, kind of physically impossible, as we know, from elementary quantum mechanics. So somehow you have to, you don't quite, in principle, it's unclear to me how it works. It's some quantum mechanics beyond my comprehension. Right? You see, usually quantum mechanics, you have a mathematical thing, when you want to apply it, you don't know what to do. There is the equation, Schrodinger equation, so you write this equation, but how to interpret it in this picture? Certainly, I have no idea. And that's the problem with my education, and I think it's not only mine, of mathematical education is not adequate to understand this, not understand on a deep level, but superficially what the hell happening there, what this arrow means, and how you can manipulate with molecules this. And on the subtle level, we cannot do it artificially. And as Feynman said, if you cannot do it, you cannot imitate it, recreate it, you don't understand it. So we don't understand how, at least maybe somebody understands it. People don't understand it infinitely more than I do. My level, even I don't know what I don't understand. I just cannot fit just energy balance. But anyway, that's a process, and then there is, when the second stage, absorption, the last one, it's another protein involved, rubisco. And so here it is, picture of that, or active site. By the way, both of them use magnesium as a metal. This when you do this kind of complicated things, energy being involved, metals. So some use there are metal core in these proteins. I don't understand why. It's always like that. It's kind of logical metals. Kind of when it comes to electricity, of course, on the molecular level, everything is electricity, electromagnetic. Electromagnetism, metals plays a special role, but it's also very ancient enzyme. Probably is older than chlorophyll, and it's very bizarre enzyme. It's very non-efficient. It works very slowly, less than 10 kind of reaction per second. Usually enzymes work some of them work in millions or in thousands. It's very slow with high level of error, and with producing some undesirable byproducts, and certainly. And again, there is long volume explaining why it is so. This kind of evolutionary explanation, or for some reason it's impossible. So some say, well, we just locked. He tried to do something and just sits in a local minimum of evolution and just can't get out of there. And some say, well, otherwise it's impossible. It wouldn't work otherwise. If otherwise, it would be not it's kind of perfect, but under given conditions. So this will briefly look at the articles. This is how it is. But again, I just want to say this: we depend on these two proteins. Our life depends on them, and we don't understand them. And this is not good. Yeah? I don't feel bad about that. Yeah. So the bread which I eat, I don't understand it. How it was produced by these two processes. Not to say about the. And this is the most abundant protein on Earth, apparently. This, a few words about it. So, of course, I just took it from the internet. I haven't read very carefully, but this, I think, is a consensus about these two proteins. So there are maybe 2,000 of fundamental proteins which you used every second, many billions of times, and our life depends on them. Now, what comes next? This is about photosynthesis. And now, next story is, which I start, because with photosynthesis we don't participate. Humanity has nothing to do with that. So we use it, we exploit it, we use it in plants, but we have no kind of interference in this, no more than, say, ants or no, bacteria. We just use the product of that, and that's it. But as for production of nitrogen, it's another story. It's much easier, simpler process, because they had to use light and chemistry and do something. And you see, it's still well beyond what we can do, yeah, now technologically. And this was always a dream to make something like that artificially. But this is far from modern technology. But making chemical reaction just using very kind of simple energy, just temperature and pressure, and this was bringing kind of neutral nitrogen. And you see, even the name of this says neutral. Nitrogen is not a neutral element like argon, but it's still rather neutral because the connection between two atoms in nitrogen, it's 3 bonds, and it's very strong. It's about 1,000 kilojoules per mole. It's certainly one of the strongest bonds in chemical. And breaking is very difficult. However, this enzyme does it, and the enzyme can contain molybdenum. Right? See, it is containing molybdenum. Is kind of, and this is a kind of describe what's happening there. It's complicated also process. There's metal there, and there are lots of ATP. And I don't understand how this can work, yeah, because ATP is tiny little bit of energy. How they produce? You need some strong photon or something to break these bonds. I just don't know this level of chemistry. I wish I knew it, but it's something which I think. If you ask me, I have to say I don't know how the hell this works. There are this picture, there is this enzyme, you can see how big it is and what it does, but I'm pretty certain most biologists don't know it either. Yeah, it's quantum chemistry, and you cannot just look at this, understand it. You have to learn it, and it takes a long time. It's not, you don't have time for the moment for that. But then there is a much easier process, an industrial process, and easier to understand. And this is a machinery, and so how it works. So if you look at so how it works, how these bonds are being broken. So if you look at the product and what goes out of that, you see that it is, you gain energy, so to speak, yeah. So you go energy-wise downstream, but there is a potential barrier. And again, this barrier, when people say the potential barrier, is kind of a metaphor. It's, they make this picture, probably seen many times this picture, but I think it's a very confusing picture, so you have to go, you see it there, and you have to go here, to the lower state, but you have to jump over some barrier. But you see, it is not that simple, it's a multi-dimensional system in this complicated shape, and so this barrier has not just one dimension. So, and this is how catalyzers work, not that they give it some kick or whatever. But in the case of this particular thing, I think logic is very simple. So if there would be kind of no catalyzer, what you have to do, because you see, you gain energy-wise. On the other hand, you lose entropy-wise, because out of 3 molecules involved, you produce only 2. So entropy goes down. So if you look at the Boltzmann equation, you see that still you have to, as far as free energy is concerned, so when you use probability also, so probability going from one state to another is rather low. But when you make the pressure higher, so the things become closer, so entropy goes down, and then under sufficiently high pressure, this may happen. Secondly, in order to do something, you use higher temperature. So what happens with high temperature? You still have very strong bond, but now some molecule move very fast, and this very fast accidentally, and this very fast moving molecules can hit along the bond, and then it jumps over this barrier. Right? That's the logic. That's okay. This we understand. When it happens in one cell, in this, on the level of this enzyme nitrogenase, somehow I don't quite understand it. When having in bulk, we understand where the energy comes. When if photon involved, it's because even less clear how it works. So, but you need some moment, you need to produce some fast-moving molecules, moving so much its kinetic energy overweight that, so it must move. It's not that bad because energy is quadratic here in the speed of molecule. If temperature goes doubles energy. No, energy is energy. Actually, still only energy. So it might be quite, but the energy, the molecule haven't moved terribly fast. It's faster, but like square root of energy. It's one point. But secondly, still probability of this heating is very low, and then you need catalyzer. But with metallic catalyzers, like here, if I understand correctly, they are very simple. The logic of them is very simple. Still, I'm not certain I fully understand it, but superficially, there is no logical contradiction. So what happens, they have some affinity to the molecules involved. So some molecules become stuck to the surface of a metal. And when they're there, their local density becomes by order of magnitude higher. So you have density, the usual process goes under 200 or something atmosphere. But the density corresponding, when they sit very close, is corresponding probably to 100 times more. It corresponds many 1,000 atmospheres sitting close. And because they are close, probability of them hitting each other, their contact become much higher. And this is how this works. Of course, for usual, for enzyme, not usual, but for organic, it's much harder to explain. This is a very simple logic of catalysis and how it was used in chemistry for the last maybe 200, at least 100 years, or something more than 100 years. But for biological enzymes, I don't know the simple kind of picture. This is convincing, and this actually can make computations see how much is stuck in this. This is that doesn't require anything but elementary kind of arithmetic to believe into that. But what happens in this enzyme is this interesting using molybdenum. Molybdenum is rare but not terribly rare metal, but some of them use vanadium, and vanadium much rarer metal, and still used in there. So these micro elements are crucial in the soil. So if you don't have them, you cannot make this. So you see, each molecule needs at least one atom of either molybdenum or vanadium, so you do need them. So these rare elements may be crucial, and also they're needed for us also. And then at some moment I will say there's a problem with soils. We don't know, it's very poorly studied. distribution of these microelements, which may be crucial for the human health. Yeah? So you may have in some life, some people are not very healthy because there is no molybdenum in there. And, you know, but of course in excess all these things are toxic. Yeah. So come next, so it goes after that. Now 2 words about phosphorus. So make a picture. No. Here it is. This is not so interesting. I just say a few words about that. The major resources on phosphorus are in Morocco. At the moment, most of the phosphate, not phosphorus, but phosphate, produced in China. And at the moment, there is no shortage of that. But its resource has tendency to be dissipated. We use it, and there is limited resource. And as the way it sees in 100 years, we'll run out of phosphorus, maybe 50 years, depending how much we can do it. And then it may become an issue. But so far it's not that bad, except that we don't use it properly. And just, the same, by the way, happens with nitrogen, with ammonia. They're not used properly, lots of that's getting lost. And when it goes to an environment, it has tremendously strong effect. Because, as I said, with bound nitrogen, we produce as much as all the nature does. So if it goes to environment, it's do something tremendous, and it does happen. It changes ecology, and sometimes in a very unpleasant way, right? Because some particular plants, say, or usually microplants start developing, and they suppress other life. So this is one of the big problems with nitrogen, as serious, by the way, as with carbon dioxide. But it's rarely mentioned for some reason, though we see it results much more. For the moment, it's much more damaging than excessive production of carbon dioxide. Now look, the other thing I want to look at energy-wise. So what we do energy-wise, and this is interesting. I want to see this picture. Yes, it is. It is energy, right? So we produce how much? This goes in terawatt hours, 160,000 terawatt hours. How much is this? So watt second is 1 joule, right? So again, if you have all this definition, it doesn't tell you much. So so what computation I can make, I made is, I think it means it's every day this amount of energy corresponds to 7 million meals. So if it's how much you eat, say 7 million meals, its energy corresponds to that. I made this computation, but I'm not certain I could have lose a couple of zeros. There are so many zeros. It's certainly easy computation to transform calories, watts, etc. But the big numbers. I think it's correct what I'm saying. Yeah, my computation about that. That's how much you produce. Right. Now, what, by the way, corresponds. That this see the tiny part of what we receive from the sun. So if you compare to how much energy we have from the sun, it's probably 1,000th, less than 1,000%. Or no, it's, I think 10,000%. So much more, of course, energy comes by the sun. The sun, I believe, delivers you your meal every 2 hours. 1 square meter of the sun, if you direct sun, give you the amount of energy which you need to eat in 2 hours, what you have to eat. Now, how much you eat energy-wise? So, what does these calories mean? Yeah? So, how much, for example, temperature, much water you can warm with for example, your own body. So this lifts your temperature, again, roughly, if you per 100 kilograms, by 35 degrees. So 2,000 kilocalories. A little bit depends on how you are not quite water. So it's easy to eat, to do only half of the water specific heat property of your body. And how much you can go. This is more or less the amount of food and energy to leave you to the Gemalungma, to the highest point on the Earth. Which means if you jump from there and go in the vacuum and hit the Earth, your temperature of the body goes by 35 degrees. Yeah? So it's symmetric, right? And physics is very time symmetric. These are kind of scale of the energy involved. So it's a tremendous amount of energy, of course, compared to what we actually need to survive. It's million times more per person. And again, there are many other scales which you have to have in mind. This is one of them. When you see on this table and see these numbers, it's very hard to understand. Yeah. By the way, without nitrogen, again, just computation you can make, you have about 25 kilograms of nitrogen per person being produced. I mean ammonia being produced. Yeah. So it's a significant amount. It's just, but in your body, you have about 3% of it. Yeah, of nitrogen. I don't know how much is being exchanged. It's certainly being exchanged. Yeah, you lose your skin, and this is proteins, and they contain nitrogen. But I don't know, by the way, how much it happens to other part of your body. Some of them, of course, being exchanged not so fast. But it's not needed for energy production. Nitrogen is only for building your body. So now look how much we eat. This is also an interesting table by how people eat. And yeah, but I don't know how kind of instructive with this, yeah. I look at this superficially just to show them and to demonstrate them and to get some feeling of that. It's not so easy. Of course, in different countries, people eat somewhat different food. This, I think, was made in England. And also there's something about amount of energy in different foods, yeah. That's, of course, instructive, yeah. And, of course, most energy contains in fat or alcohol. Everything else is below. Okay, forget about that, but what is more instruction? Oh, no. Yeah. Something else. This is a mass, a relative mass human take on Earth. So what happens, this ratio between our mass compared to the mass of other animals in the last 100 years changed by a factor of about 20. Right? So at the moment, animals become, well, maybe even more than that. Animals reduce by a factor of 5 and we increase by a factor of about 5. And this gives you a relative weights of what happens. And this kind of much more better understood. Table numbers here we understand. So GT means, of course, gigaton, billion ton. Plants, number one, of course, the dominant. And then goes bacteria. And all animals you see, just including human, is about 2 gigatons. So we are even less than viruses or whatever. I know viruses, of course, actually very small. I'm sorry. Viruses are many, but they're very small. This is the estimate of them. But bacteria are dominant. Now there are bacteria in archaea, and there are two kinds of. They have a different name. Archaea also kind of bacteria, but they're different kind of. They're very far removed from bacteria. They're further removed from bacteria than we from bacteria. We are close to archaea than to bacteria, but that's not the point, but that's give you a good idea of what happens. So the multicellular organisms are plants, fungi, and animals, and then there is separately table for animals. And humans also make significant percent. Of course, arthropods and insects dominates, and fish also, a lot of fish. But when it comes to mammals, there are more of us by far than mammals, and livestock even more significant of that, twice more than we. You see, we're 10 times more human than wild mammals, including, I think, those who live in the ocean. So it's a big number. 8 billion is a big number, and so this is a visible thing. And so we just, I cannot add anything, just you have to, I think, look at this table and to have give some idea of what happens on this planet biologically. Of course, it's opposite view if you look from a molecular point of view. But these things are related. And they're related to the fact, this proportion, because exactly the energy which needed to break to 3 bonds in the nitrogen is slightly smaller than the bonds in the product. Otherwise, it would be a completely different picture, at least as far as people are concerned, right? So it's actually about 5% difference in these energies, and this wouldn't happen. It's only possible that it can be industrially rather easily produced. Of course, if not for that, I think it's even maybe biologically it would be impossible to have to make fixation of nitrogen by simple process. It would be negligible amount of bound nitrogen, and life wouldn't emerge, right? So this little thing with molecules determines, define our existence, and they're kind of accidental. They're usually not said so. There are many of these accidents. Of course, what's happening in the history of Earth was another chain of accidents which brought us here, and there are this little kind of chemical stuff. And you can say, well, life would find some other way to do it, but this is just empty words. Yeah, we have no basis to believe so. We have only one life, all based on this chemistry, specifically on the chemistry of nitrogen, and we cannot imagine life without that. And this, again, kind of a mathematical problem. What level of molecular complexity you need to have life? And this is, if you formulate it properly, and that's one interesting kind of problem for me, first formulate it and then look at this, and then we can say, how realistic it has to have it on different chemistry. Possibly specific of the chemistry, maybe not so important. Yeah, this is historical distribution of animal mass on mammals. Yeah, and first, how it was carbon-wise. Yeah, we can measure the total weight of carbon-wise, and I'm not certain which tables do what. But this, as far as proportion is concerned, it's material, because its distribution more or less the same. So if you look at 100 years before and what happens now, you see proportion change tremendously. You see wild animals gone down, and human jumps, especially livestock and human lives up, gone up. And now, just looking at that, just think again in evolutionary terms what it may mean. Of course, we are not exactly following evolution, but if you take kind of formulas which are common in Darwinistic discussion, which of course extremely kind of undesirable, but the expression that the fittest survives, and there is this rate of reproduction determines the future, it's not true in a very fundamental sense that if you have some animal population and there are two kind of slightly different species occupying similar territory, and then one becomes dominant and it reproduces faster and has kind of dominance, which happens with ourselves here, or good example when it's happening when there are very large advantage of one species over another. And this happened, for example, in Australia, which followed slightly slower rate of evolution, and when modern animals came there besides human, for example, dogs and rabbits, and they took over and they certainly dominated the local fauna, like dingo, which became major predator in Australia, and the rabbits, of course, populated tremendously, and so it is huge overpopulation. But there they have nobody to compete. If you have to compete with somebody, still you may start dominant and you have much more of X compared to Y, but at some moment it may stop and have opposite effect, because the moment concentration of X become too close, they may have some virus or bacteria adapted to them, and when plotness, the density of the population goes above some level, some infection starts and wipes out almost all of this population, and then Y become dominant instead, because environment also changes, yeah, because evolution is not evolution of individuals, it's population in the surrounding of other populations. So evolution works in the terms of population dynamics, which, by the way, also highly oscillatory process. It's not like monotone function, as was imagined by classical meaning Darwinian kind of evolutionists who certainly don't understand, didn't have very poor feeling for mathematics. Knowledge, of course, Darwin, his major, from my point of view, his major contribution was understanding mathematics, not biology. I don't think he was biologically everything he was. Well, I think almost everything what he was saying was either not original or correct, but his mathematical kind of philosophical thinking was correct, and this determined the shape of the subject. And this is kind of amusing. He was mathematician at the core. And though he was not aware of that, he was complaining he didn't know mathematics well enough, which means he realized he thought mathematically. And so could realize he not doing it good enough. Of course, if you have no mathematical perception at all, you're not aware of that. Right? But he's complained about that he was a bad mathematician, in the case he was a mathematician. And now, this is the distribution of these animals, and so what comes next? Again, somehow they can stop. And then there is this, again, look at this. So this is a population table, which was before. So you see how we grow, and this rate of growth, some people say it slows down, it's negligible, right? And more or less the same, 2.3. Of course, some part of the world go down and some go up, say in France and everywhere in the developed world. Population goes down, and it goes up in, say, in Africa, or in part of Asia, and it still goes up. And so, and then there was this discussion of Malthus, following, kind of criticizing Condorcet, who knew that, of course, because Condorcet was a mathematician and politician, and was killed by revolution in France, died in prison, and who kind of said what you do with that, and Malthus said not. And what's interesting about that, it is not so much this Malthusian point of view, we don't know what happens and we have to do something without that, but the attitude, how it's being discussed, and that is this partly, in not such a horrible way, appears in every way in biology, but where this Malthusian business, because this is another thing, because on those pictures which comes before Malthusian, there was this, because you see my pictures don't quite correspond, because I don't know if somebody might explain me how to force pictures appear where I want them. Yeah. It is another issue before I come to it. It's about agriculture. So now almost a significant percentage, close to full usable land, are being already used, and this number is limited, and so there is a big problem with them, partly related to the artificial fertilizers, because what happens to plants with the artificial fertilizer, the same which happens societies when there is a well-developed welfare. If there is sufficient developed welfare, eventually society collapses, because well, people stopped working hard enough, and the welfare takes kind of helps those who don't work. And the same happens with exactly the same logically, mathematically, the same happens to plants, because normally plants, about 20 or 30% of their energy taken from the sun, they give to the other organism living in the soil, essentially to fungi and to bacteria. And this fungi and bacteria makes soil kind of keep soil in a good shape. Just by their presence, they give an organic to that, and in turns, they help the plants to absorb more nutrients, because roots are not good enough. And particular fungi which attach to the roots, they enlarge the area, absorption area, by a factor of 10. And certainly, without them, most plants cannot live, unless they have artificially fertilized. If the density of fertilizers, of nutrients in soil goes above a certain level, they don't need to do this extra work. And so they flourish, they grow very well, but the soil eventually becomes depleted of this organic material. The same as happens with, similarly what happens to societies with a well-developed welfare, and this of course what happens to all our societies, and they may collapse eventually. If this has not changed, it will not be changed, as we have examples like in Argentina, to what happened to it. Extreme welfare is destructive. And with plants the same happens, and the soil, when it loses enough organic material, becomes unstable. And its substance, the structure, mechanical substance of the soil become different, and because easily erosion of the soil goes up by a factor of 10. And certainly at the moment it's not so bad, and we don't think we have felt this out of erosion in the last 20 or 30 years, contrary to some prediction. But it will not completely full erosion, but significant erosion. And if it happens without control, then again, billion, in 30 years, maybe mere 50, 500 million people will be without food. They will be not able to produce their own food, which actually already happens in most part of the Africa. The soil, the level of erosion, if the people cannot produce their own food. So they need to import food from other countries, for example, from Ukraine, which now under the war, it may happen that if the war goes on, the soil will be not usable for some period of time. It's not felt now. It's still, there is even overproduction of food. But this is just a moment. Yeah, the more you produce now, the less it will be there, because we use a resource which is not, we don't know, soil is soil, you can't replace it by anything. And so it's a big problem how to change it. Actually, even, so if you don't use artificial fertilizers, the quality goes down. Of course, there is natural, processes which bind and fix nitrogen, but they're also not without price, yeah. So, and it is so it's not understood what we should do in this respect. If you don't do anything, it will be pretty bad. This is another situation. We know if we continue the way we are, that will be disastrous in many respects. The question is, there are potential directions to do it, but we don't know them enough, and particularly in the soil. It requires more study to decide what to do with soil. And this is actually, when I was thinking about lectures, my major emphasis was to learn more about soil. But this is impossible. To learn is a huge subject, and it's much easier to learn, from something like evolution, general principle of evolution, mathematics of evolution. Yeah, it's easier for mathematician to understand. And soil, you have to dedicate your life to that, to know roughly what's happening there, and still, overall knowledge, as my understanding, insufficient. Soil is very complicated. For the reason it's the most heterogeneous system on Earth. It's ecologically have the most diversity of ecology than all other part of the Earth, because it's so heterogeneous. And it is quite interesting, but, well, we don't know how to systematically study it, not in detail, but globally, and to make decisions. So the soil is a big problem. But now I want to say a few words about Malthusian problem. And so what is, wait 1 second. They had some lines. See, it's confusing because these pictures, this, by the way, another useful picture. This is a pyramid, who eats whom, right? It is so-called trophic pyramid. So there is one major network joining all organisms on Earth, who eats whom. And this is a kind of interesting, by the way, graph, and it has certain structure. If you look at this carefully, it somewhat reflects some tree-like structure, apparently reflecting the tree-like structure of evolution, which is not exactly tree-like, and that's only up to some limited extent tree-like. But this is also interesting table, because each time you go to the next level, we lose 99% of energy. So it's much more efficient to eat grass than to eat meat. And when you use this animal who eat grass and then we eat this animal, it's still not a very long chain, but the longer the chain, if you eating predators, for example, would be extremely non-efficient. For that reason, we don't eat tigers. Right? It's not efficient energetically wise. This may be regarded as an explanation. So, but this is what I wanted to show you. I want to discuss, to show you something I'm using rather unpleasantly. Yeah. Yeah, this is something about this degradation, but I don't. But it's kind of tricky. I'm not certain. I'm not certain it is correct because there is lots of information in newspapers, which is kind of just to making it impressive. Situation not as bad you can read if you read some kind of. It may look worse than it is, but it's bad. But what I want to say is something else at all. So somehow you see it is about Malthusian, yeah? Ah, yes. So the language which is used by people discussing this Malthusian overpopulation. We see the people who support Malthusian, they apologize for oppression, overlord empowered to do the necessary constraints, etc., etc. So you see, they use this kind of language, certain group of people, and we sometimes hear that, and when I hear it, I'm really scared, because, by the way, this is said by a person who is an engineer, and he was actually interesting project about kind of cosmic engineering, and he not kind of just journalist, but the country from where I came, people who said this next we would do to put the concentration camp. People who use this language, whenever they have power, they kill you or enslave you. They don't kill you immediately, they kind of try to change your opinion. First you're putting you in prison, concentration camp. It is very strange to me, because Malthus was a mathematically minded man, and he realized this exponential function, and it is a problem. And we do have this problem. If we grow at exponential rate, and which happens to many populations, eventually it will collapse. And it grows, and then it may collapse because it loses stability, something goes wrong, and we don't know what. We are not supposed to exist in such a density on this planet. And it is a potential problem. It cannot be. But that's kind of a language being used when people start speaking about that. And I said it's interesting, you know, when we speak about population, not only just already evolution of animals, also lots of things happen basically because Monod, one of the great French biologists, said the problem is evolution. Everybody believes he or she understand it. And of course and also people think they understand how population grow and so what happens in the future, and again become very emotional about that when somebody disagrees. But we don't understand, we don't know if there is a slowing of the rate population. Of course, some education apparently has this effect, but it may or may not be realizable. And what happens in certain, well, there are some sociological factors which are out of control. And this is unclear if this is not a problem. And the overpopulation is a problem, and people want to deny it. But of course, what happens is a nice part of the earth is occupied, and then people move to less pleasant areas, and people some suffer, and overpopulation may be a factor. And it's hard to say, yeah. And so when people argue about Malthusian, they argue about multiplication table, essentially. This, by the way, not very much true about so-called Darwinian evolution. When people speak about Darwinian evolution, saying that it's so always true, basically because multiplication table is true. So Darwinian point of evolution is counting numbers, and that principle of that, if I is bigger than B, then A minus B is positive, roughly, of this level. Oh, well, it's never, we see it everywhere, great theory. I mean, it's great theory that you can see world in this term, biological world, you can see these numerical terms, and this was point made by Darwin, but you cannot disprove it. There's a big difference between kind of it was Lamarckian theory, which was in many, again, I think, in a way disproved, in a way. I mean, depending on what he said. The trouble, by the way, is, say with Lamarck, I think nobody in English-speaking world ever read Lamarck, and people read sometimes incorrect translation, or sometimes translation not of Lamarck, but article written by Cuvier about Lamarck, and Cuvier was very much against, and he was kind of writing satirically and making jokes taken seriously by some writers and still being cited by people and saying, oh, well, we disprove Lamarck. I mean, this is sticky. Myself, I never read Lamarck, so I cannot say it, but this is what his opinion is. And therefore, if you don't know the originals, also as much as, say, for Darwin. Darwin, of course, we can read, and usually what being ascribed to him is not what he was saying. And then there is this kind of this idealization. We make some standard, and again, you cannot kind of touch a subject. It's not as bad as Malthusian, and actually Darwin was influenced by Malthus on the allied. There is exponential function. It grows very fast, and when there are big times and big population, many things may happen. But the major points of Darwin scientifically were conjectures which are not verifiable because they refer to large time scales. So, but now we finish with that and turn to the last topic. So what I was saying up to that point, speaking about this human part of the nature, our role was no different from any other animals, which like rabbits in Australia. We just come to a situation with some advantage, or like dingo, and for some population, and start reproducing uncontrollably. We can be consuming a lot, and everything we're doing, the way we are influencing life, like livestock production, in a smaller scale, we're in animals, like ants. We're cultivating some plants or food fungi, more or less in the same way we do, and it's no big difference. But now, in the last years, we learned how to modify life, not just domesticate, and actually using artificial evolution, which is completely biologically different from natural evolution. The analogy is incorrect, and it was realized actually by Wallace, who was contemporary with Darwin. He was pointing out that there is fundamental difference. Domesticated animals are reversible. You can put them into the wild like pigs, and pigs in several generations will become exactly looking like wild pigs. But once some species change, it doesn't go back, right? It's because on the molecular level there are different mechanisms. And Wallace actually was understanding this much better than Darwin did. But anyway, now we can modify genes. It's still not perfect. It's still, and I say this, we still didn't on the top of that, but there are steps, and it may happen in 5 years, and this may reach the point we shall be able to modify human genomes. And the moment we can do this, we can produce people the way we want them, or somebody want them, and this will be a different world. You see, unlike artificial intelligence, because their tempo of development was very, very slow. After Turing, there were no, or maybe some immediate work after him, there were no qualitatively new ideas or results. Quantity grew tremendously, power of computers, a number of people involved, and the technique of programming techniques, but not kind of big progress, at least for the last 40 years. If you talk to people who were in the field, what they say, there were no progress. I mean, except for growing in the volume. And with biology, it's completely different. Yeah, we have significant steps every 5-10 years. And therefore, in the coming 5-10 years, there is a quite realistic thing. We shall be able to modify human genomes on the human embryos, just in the cell. And then, of course, we shall have a different human population, or part of them. And in particular, those which will be able to solve like language problem like that. Yeah, I mean, have 10 times somebody smarter than Ramanujan was, and so it will be solved, right? And this is how we have to solve it. You know that how long it takes for, if you take monkeys to working on a printer to produce Hamlet or Shakespeare. Of course, if it's just modern monkeys, it may not work, but if you take the predecessors 6 million years ago, and they did it, yeah. But except you don't have to do it typing all the time. You have to reproduce fast, and that's it. And the same is here in order to solving this length problem. Maybe you don't have to think about that, but you have to think how to modify human genome. And then it will be on the core. You understand the mechanism of proving that your brain can be improved, and can be improved without really understanding how it works. You just start making game with that, and you shall do it. So this may be the way you have to solve the mathematical problems. So see how it's done. Yeah, this is, I think, the most interesting. There are two aspects of what I was saying. One interesting thing is it's fun to look at molecular biology because it has beautiful structures and some logic in them, and especially it's easier to talk about them when you speak about evolution, because everybody, I was included, can pretend we understand it and say something clever about that, especially finding mistakes in what other people say. It's easy. And then if you look how we ourselves fit into the ecology of the world, it certainly sounds very pessimistic, because we destroy it, we do it blindly and keep destroying it, and eventually we kind of, the same as actually we do with our society. We're sitting on a branch which we're trying to cut, and we're cutting it without understanding what we're doing. It's not good. However, when we look at the last thing, at least we understand what we are doing. We can modify life. We can really effectively channel it in some direction we choose. Whether it will be right or wrong, it's hard to say, but at least it's not blind animal behavior. At this moment, to this point, human behavior was biologically the same as of ants, no more, not so matter. In a way, I would say that, of course, the advantage of human exactly that we are not rational. Unlike insects, our behavior is irrational, and for that reason we moved faster. But now this is irrationality, like understanding cells and things like that was for pure fun, brought us to the point we can change life, and change ourselves. And this is the third part, which makes you optimistic. Or at least something new happens. It makes we change our direction, because we do know if we continue the way we are, we shall be obliterated in 1 century at least. But now we can do something else, so we have a chance, right? And this is one of our chances. And so where it starts? Of course, there were many discoveries, and the one of this PCR. This was, I think, some turning point in biology. It was several points, but one of the points which make biology the way it is. A tremendous technological progress, using biology to making, kind of cheating a little bit your biological system. You force it to do, it's not meant to. We behave like viruses in this respect. Like virus goes to the cell, it knows how to push some button, and it does something what virus want to do. And humans manage to be as smart as virus. So at this level, we repeat, we're going to the bottom of the life, the viruses, and try to do what viruses do. And then, what is PCR? So, namely, forcing biological system works for our advantage. And PCR is a process when you have one molecule, literally maybe one molecule in a bowl, when there are a trillion of other similar molecules, right? For example, there's molecule one of them, of you, and where there are other molecules, the DNA taken from somebody else in billions, and yours only one. However, we can extract this one. Not exactly extract, but at least to know it is there, right? And that's the problem. So we have a trillion identical objects, and one of them slightly special. But how to extract it there? It's there, but you cannot kind of sort through 1 billion units, or it may be more than billion, maybe trillion, of course, easily. But even when it's million, you cannot sort there to find small object about million almost identical. But what you can do, you can amplify this special one. You can force it to multiply like animals do, and then at some moment this overwhelm and become majority in this community, and then you can see it clearly, right? And so how it works. And so what we exploit, the machinery of DNA replication. There is fantastic machinery in every cell. There is a machinery which replicates DNA. There was one DNA, and there is a process which does 2 DNA. And this was understood pretty well at the moment how it works. And it also was realized we can produce some artificial process which makes it work even better than it works in normal life. Because in your cell, one of the major problems, we have 2 strings DNA. And in order to make duplication, you make a template to each of them. It's actually templating a relatively easy process, but you have to separate them, right? You have to stretch them like that, and this is a complicated process, and there is special enzyme which does it, and it takes actually energy to do that. And then kind of one polymerase goes in one direction, another direction. It's a complicated process, but the major thing is templating one. And then, but you say, but if they separate to start with, it's much easier. So if you have one-stranded DNA, it's very easy to make it, and you have to trick it to do that. And you know the trick because you know what happens, what you have to do. So let me show the picture. So you use what you know about this process. It's in itself, it's complicated, but you do something artificial to make it much simpler. And then this miraculous process happens. Miraculous because you really solve a kind of impossible problem. So how it works? So one thing, you have this one-stranded molecule. In order to be replicated, in order to this polymerase produce duplicate of that. It must come to the right end, and that on this end you attach little piece of another DNA or another RNA. Typically done in cell with RNA, but you do it with DNA, and this enzyme doesn't notice the difference. Yeah, and once you do that, so you have your many molecules, but you know the one molecule which you look for. You know what it is, what's written there, and so you take complementary short string of some molecule and throw in the solution. All these molecules, and first you warm your solution. It becomes warm, and then the strands go away. They kind of, it's like a kind of rigid thing go to the liquid. And melting. It's called melting of DNA. It melts, and the two things. So temperature goes for about 90 centigrade. Now they're one-stranded ones, and you throw the huge amount of the short pieces complementary to the one which you look for. And then one of them stick to the right place and completely ignores the one, because you see, if you have, because it's binary code, if you have already 30 or something, 2 to the power 30 is a big number. It's overwhelmed the number of these molecules, yeah? The power exponential, and the power in positional information. It's a relatively short string, maybe 30 or 40 units, but if you take 2, or actually 4, to the power of this 40, it's a very big number. It's more than the number of other molecules there, right? So use some mathematics here, not simple, not hard, but it's on the Malthusian level. You make computation of big powers. And then when you have these molecules, and there is this kind of thing attached to that, then polymerase, and you throw there many polymerase, it will find it by random moving, yeah, but you throw a sufficient number of polymerase and start working. By the way, this polymerase need ATP, so I have to throw also ATP. And this is what we see in this picture. Yeah, so you see this. So you see, you add this primer, that's called primer, it sits there, and after that, polymerase starts working, and this becomes double. We have complementary strand. Now we have complementary to each of them, and then they may come together, so we have a new molecule which we had not before. Now you repeat the process and keep repeating it, repeat and repeat. And at each round, you double the number of molecules. So if you do it sufficiently many times, then you have a huge number of these molecules. The one, you see, the primer will find only your molecules, yeah? Because a short string already determines everything, then, right? Because the 2 to the 40, it's a big number. You don't need to know the whole molecules. You use the whole information, this molecule, only this short string. And this is how it works. There is one point here, and you have to use polymerase so they come together, systematically melted, and you have this temperature varying there. And usually polymerase doesn't work actually in this high temperature. And in order to work, you need special polymerase. It was again quite remarkable. It was discovered. There are bacteria living in very hot waters and about more than 100 centigrade, and there are proteins which can work at this temperature. So it's again was remarkable. This study of this exotic bacteria was crucial for that. And now you know this PCR used everywhere, the most efficient test for everything. because you can find particular string of DNA in a tremendously kind of uncomplicated environment of similar ones. And so it was again discovered due to purely scientific research, which is a rather rare thing, yeah. So this, you do think, there are other things in biology were done purely for fun, and then had this incredible effect on our life. So this is one of the first technological, I would say, quite simple technological thing. There were other one, but this was kind of the very pronounced one. And this I'm using history. The guy who discovered it done it under the influence of LSD, and well, there is kind of controversy who done it first. But this is not so. It's still interesting. Second thing is CRISPR. Again, I say in two words, but each of them is a kind of big subject and not at all simple. So what was discovered at some point that bacteria, it was done by bioinformaticians, I think. There are particular patterns in genomes of many bacteria, some repeating patterns, and which was kind of palindromic sequences appearing, and the people couldn't understand their role, and so what was happening. But then they realized that some pieces of DNA between the species, between these two inverted strings, were DNA from some viruses. So they found in bacterial genome some patterns from viral DNA, and they realized, and so several people purely theoretically predicted that this must be a kind of immune system for bacteria. And by the way, the moment you look at that, it completely changes your perspective on the kind of, there is Darwinian evolution, Lamarckian evolution. It's rather absurd terminology which doesn't correspond to reality, but it's shown that the canonical view, or kind of that the reason of Lamarckian evolution, whatever it means, was completely false. In bacteria, major steps of evolution are Lamarckian. So, and because there are some mechanisms which we can implement it, which is immunity in bacteria. And then using that mechanism in bacteria, which what I was doing, I don't have time to explain, it's, again, logic of that is simple, but of course implementation of them took years of very smart people, and they modified it. As usual, it doesn't work the way, it's not exactly how it works in bacteria, but slightly modified, actually simplified it. And what they managed to achieve modification of any given genome at any given desirable place, to cut it and replace it by something else. But this is much better by order of magnitude. It was something like was done before, but now it's done by 2 orders of magnitude better, because before the error in a long process, and error was about 99%, and now error, I guess, about maybe 60%. Still more than half, I guess. See this? I looked at the literature. It's hard to, it's lots of literature, so I look at this superficially. But my understanding is the problem with this process. In principle, you cannot do it with a high degree of fidelity, any of this process, unless you use energy. And this process doesn't use energy. And when you do it to the virus, so the purpose of this making the virus, when the virus enters the cell, then this immunity sends this message, it's stuck to a particular place in the virus, and then this protein which cuts the viral DNA and kill the virus. And if it does in the wrong place, who cares, it still kills the virus. But if you want to make some modification, you have to do it precise place. And when you do experiments with animals, okay, you do it and just some animals die, it's okay, but then one we get, it's okay, to have modified animal. But you cannot do it with human, you see, that's the problem. It must have high fidelity, it must be close to 100%. And actually, there were some big problems with the first genetics modification, trying to modify human genome, people died. Not for that reason, but for another reason. Not because of the mistake. But this can be used to modify your genome at the beginning of in the egg. And for example, insert a piece of DNA which you think is maybe advantageous. And start making such experiments, for example, making people smarter, stronger, and more. And this is ahead of us, but you have to couple this with ATP. And this is tricky. And so you have to make modification of this process. So already I mentioned one thing in other process as was used for the modern vaccines. So I showed that and then showed the last one. No, the last, so this is, no. Yeah, this is a comparable thing which is done with on the comparable level of sophistication, how modern vaccines are done. So I don't want to explain what it is, but you see, you have to know in a very well how your cell functions. You need to know this dynamics and structure of the cell, and then design this vaccine with using RNA. It has some similarity because it uses RNA in both. RNA play a crucial role in information written on this RNA. So you give actually a message. You not just put some drug or whatever, but you convey some message to your cells. And they do the same. You convey some message, and the message written on RNA. And so you can talk to the organism and tell him, do this or this or this. You really understand this language. Yeah, it's fundamentally different kind of medicine. You don't, all other medicine up to more like having something like a hammer. You just do whoop and see the result, either you break or not. But now we can tell the cell, do this or not that. But then something you can do and something you cannot do. And then how you can achieve new things. And then comes the last, not the least, discovery of the last decade. This is a phage controlled, phage-directed continuous evolution. So now comes the evolution, of course. Yeah. And again, people say, oh, well, something has to do with Darwin. Certainly not. It's more or less like breeding animals. You want to breed animals with some purpose. But now, it still works in a very primitive way. So amazingly, so what was a great improvement it was earlier, but still it's not the last step of what can be done. So how before artificial evolution was? You want to produce bacteria doing something. Just this process, by the way, with CRISPR allows you to modify gene in bacteria in a desirable way. But another way is to breed bacteria for a certain purpose. For example, you have one enzyme doing some purpose, and you know there is another potential function similar to that. It digests some X, and there is a modified X, and you want to digest it. So you start modifying, you throw bacteria in some medium, and they survive whenever they do things right, and they survive the better they do it, and so you making the cycle, and it's a tremendous mess. People were doing that. It works very badly. Sometimes it does, but it doesn't work so well, because bacteria don't like this manipulation. They don't kind of cooperate with you. But anyway, it's rather primitive, because your mutation, which you produce just by noise, and bacteria resist. But this was a great improvement. And the idea is you make this selection and mutation in viruses, not in bacteria. So what you do, you make some part of the genome which control behavior of the virus, put it into the inside of the bacteria, and then, but what you want to is modification of some other enzyme which is bacterial one. So you exchange kind of these things, yeah. And so, because viruses reproduce much faster than bacteria, and they cannot adapt that easily because they don't have complicated function. So you use bacteria just as a medium, as environment where the viruses live. So bacteria, the flow of bacteria, and viruses evolve, but they all the time interact with bacteria. They don't kill them, but they go back and forth. But so there is circulation, and viruses move. The replication rate is 3 times more faster than bacteria, and which is moreover they're much easier control, yeah, than bacteria. And so you can couple some bacteria, some gene in production of some proteins in bacteria and necessary protein in the virus, and you force bacterial protein to be modified and mutate. Adapt faster using viruses rather than bacteria. And because you then couple them, you have tremendous flexibility. You don't have to be advantageous in any way to the virus or the bacteria. You choose your protein you want to modify in bacteria, couple it with some virus protein necessary to the virus, you put them all in bacteria, and virus forced to modify bacterial protein in order to survive. And then a remarkable variation of that developed a few years ago, which is done there. But this again, you see, it's still a very primitive system, because the only mutation which happen, they are random. They are just random noise, yeah, and they certainly don't work very efficiently, right? Still, they work not so bad. They work by far better than probability. If you just compute probability of this working, you will see it will work hundreds of years, and sometimes it works in a matter of weeks, yeah, but still not very well. But on the other hand, there is another way, just modifying by this CRISPR other system, and of course you have to couple them. So this is what will come next. I'm fairly certain in biology. You couple these two mechanisms, this kind of evolution, which will be certainly refined further, and CRISPR-type modification, and then it really starts a new stage of this stuff. So you can produce anything you want pretty fast, any kind, including, and then you can couple it with probably with eukaryotes, with animal cells, and then it will be kind of new phase of the development. I think 5-10 years we shall see it, and that will be fun, and this we can participate. Yeah, you can imagine how scheme, this is combinatorics. Even here there is some combinatorics, how these elementary processes which cell can do, and bacteria can do, and chemical can do, they're all there. You don't have to truly understand very well how they do. You have to only know what they do and understand it in detail, so not to make mistake. And then you can combine them, like you make a computer out of chips. Yeah. So these chips already there, they're in life. You have to extract them from the knowledge there, and then you combine them, and then you produce new machines, biological machine, and then may change our life for better or for worse, we don't know. But as I think is rather clear, if you don't do something radical, we have very bleak good future. If you systematically look what is ahead of us, it doesn't look good if we continue the trend we do. It certainly applies not only to science but also to our structure of our society sociologically, where unfortunately there is no such solution. But that will be fun, and we come to the new world when we shall be able to modify ourselves. And then, of course, there are many science fiction stories about that, and we come to that, and this will be, and as a mathematician, we can be part of that. Okay, so this is the last I wanted to say. Unfortunately, neither had enough time to learn it more, even what little I learned, I have no time to explain it in details how it works. But the logic of this particular one is really remarkable. You take bacteria, you take virus, exchange their two proteins, and force this evolution. And this is a very mechanical process. Yeah, you just don't have to interfere. You make this machinery and runs by itself. And of course, on the next level, you have to combine it with genetic manipulation and make a computer controlling all that, choosing the right modification to make, not to make it to the virus decide, or not to make the cell to decide, but your program will decide. So you make this system, and this is all within modern technology. It's just a question of will to do it, and I think it is an obvious thing to do. I mean, which is. Of course, this to do it, you see what's again in this, like in the previous occasion, it depends on biology to do it on the basis of their kind of understanding of what happens in the cell. And it seems to me that the present day, if you do make any suggestion which is not absurd, roughly corresponding to what cell can do, experimental biologists can do that. It's a tremendously developed kind of technology. Everything possible, of course they cannot go against the laws of physics. Sometimes, some biologists suggest things which contradicts the second law, and you cannot do it. Yeah. Strangely enough, the physical laws should be observed, but apart from that, almost everything is possible if you do it correctly. Okay, so in my lectures you may ask me something, and of course most of the question I will be not able to answer, because I don't know the subject matter. I have only amateurish knowledge of all that. But it's still interesting. So, we'll see there's 3 different topics. They linked in a kind of bizarre way, just when kind of basic molecular biology mathematically looked upon, what that tells you and what the consequences it has to our life and our participation of ecology as animals, not as intelligent being. And then, so it happens when our intelligence came to the point which may overrun our instincts, so to speak. And this is three phases. The understanding the first changes our position in this world, where again, I keep repeating, we still behave like no smarter than any kind of ants, yeah? Because we are driven by, just especially looking at this discussion when people speak about Malthus, this exactly, I think it is a not joke that the part of the brain involves the limbic part of the brain. And when you look at that, my limbic part immediately switches off. You know what limbic part tells? When you go and step into something you don't want to say it, then you say it, yeah? This is the limbic part works. And this is a dominates. Whenever it switches works, it dominates your cortex. And this is a critical situation, exactly this moves us, not so-called rational. Cortex, of course, is not rational. It's certainly irrational. But still your cortex can now, via molecular biology, probably will be the major step in evolution. So it's not a joke. Yeah, we're going to the next step of evolution. We entered Anthropocene, and the beginning of that was just very simple chemical reaction in more or less 100 years since, and now we're going to the next step in our. So see, but if you look at the growth of us during the whole time, and then it was really boom, look like that, and then it goes like that. And now something is going to happen. It will be really new. And we may be destroyed before that by some simple kind of physical mechanism. But if not, we shall start a new species. Biologically, we shall become different species, radically different. Yeah. But of course, it will be not so easy socially. But this we're coming to that. And now there is the memorandum, people start saying, oh, we shall make some rules not to do that, but of course it will be done. The moment it will be possible, it will be done. And 100,000 million of you newborn children will be different from us. Everybody 10 times smarter, for example. Yeah? It will be very bad for those who will be behind. Right? And this will be certain tremendous strain on the society, but it's still more interesting than just, slowly dying, which certainly would happen otherwise. So this is on the optimistic kind of line. I will finish it here, because for the rest it's certainly very depressing. If you look at the ecology and sociology, it's rather depressing if you extrapolate. Again, it's extrapolation very easy. You don't have to, because it's never word that we are counting in the part of the earth being used. Yeah, you see, we're using kind of 60% of what is there. It was never like that. Yeah, it was maybe 6% 100 years ago, and now it's 60, and it's complete. But percentage it's not so this. We're close to the maximum. It was never like before. People say, "Oh, well, it was like that before." It was never like that before. We never had this exponential. And whenever it happened, some animals, we know it always collapsed. Never, it can go for a very long time. And it's very short time, of course, of the existing. So there is no past is different. And say, if you look at particularly what happened on the level of biology, never ever close humans were so much advanced. And this is not quite appreciated. Now people speak about this vaccine, whether it's useful or not useful. This is such a trivial matters. By itself, it's an incredible discovery. It's a fantastic kind of jump in our knowledge. And this is not, I think it should be, a mathematician at least should be able intellectually appreciate this what's happening in molecular biology. And this has started, well, about 40, 50 years ago. It's rather recent. It's rather recent, yeah. So basically, it's a propaganda for this remarkable development in human culture. Because you see, in artificial intelligence, I must say, it was a big progress, but it was zero contribution to our culture. It's so technical. You cannot learn anything from there because nothing was discovered. It just a tremendous amount of effort was put and this will be done. But here many things, incredible number of new things we discovered, both experimentally and ideologically. We discovered so many things, and they're not so well known. And this is also will have comparable effect with artificial intelligence. Well, it's unclear, yeah, if it so it will happen to it. It's completely different in the structure. And here we have community of people, of biologists who every 5-10 years make this tremendous new steps. And this is worth knowing for two reasons. First, it's fun to learn, and secondly, our life depends on that. And thirdly, we may participate in that and direct this the way we want. This is so. Every reason to spend. If you, again, I'm not joking. If you want to have a mathematical problem, this may be the fastest way to solve it. You modify. It may be something else, of course. Maybe you can do it via artificial intelligence, and there is something to think there. Yeah. The way it is certainly rather technical, and hardly will solve deep problems. The way it stands. Maybe I'm wrong, of course. Yeah. It's easy to appreciate this, but this is something more interesting. This you learn something new at every several years. So I highly recommend it. So, but I think there is no question. You too. I don't expect any question. You may ask some question. No, nobody.
So, can I beg to ask one question?
Yeah.
You predicted the evolutions. What would be a time frame?
Oh, yeah, well, in 5 years. My expectation in 5-10 years. Technology to the point of modify human genome. And then a new generation will come and they have to grow up. It will be 25-30 years. People, they will be modified, it's genomes, yeah? So some new children are appearing. And then in 20 and 30 years, there will be definite population of new people. Absolutely, some of them, of course, experiment will be unsuccessful, but some of them will be different. And then it's, well, it can be too fast because it depends how fast people can grow. And of course, it may take different form, yeah? But the kind of obvious, so this thing, looking at the time development, in 10 years, we shall have perfect modification. And it's clear what to do. I mean, it's even for amateur, it's obvious what to do. It's just technical thing to do. You have to couple this process with ATP and then couple them with this kind of artificial continuous evolution. And this is like genome project, yeah. Genome project, its implementation take long time. Some people say, oh, well, it's not so good. Fantastically, 20 years, and we have without this genome, this CRISPR wouldn't be discovered because it was discovered by bioinformatics, and this bioinformatics based on tremendous library, on the library of known genomes bacteria, because you can make this genome now like that. You develop this technology with genomes, and then, of course, you have powerful computers and mathematicians who develop techniques how to compare this genome, and then they discovered this CRISPR. They discovered this very much like mathematically. And then, of course, it was implemented physically. And this will continue, this process. So it's pretty fast, maybe 5 years, 10 years, 20 years, but this is very short. It may happen tomorrow. Some already people working, I'm sure people trying to do that now. From which lab when it's come. It's a little bit slowed. It was discovered about 5-7 years ago, that was slowed. And the reason is, in my view, it is they cannot do it with ATP. Again, maybe I'm wrong. My naive kind of understanding of thermodynamics says it's impossible for thermodynamical reason. The way it is, it never will be precise. That so far we have no input of energy, you cannot make things sufficiently efficient. And this is a kind of, I think it follows from second law of thermodynamics. Because the cell uses a lot of ATP. They have a helicase to unwind two things. And this helicase uses a lot of ATP. And then it does very precisely, works very well. All mechanisms of the cell, which have high degree of precision, works with energy, namely with much use of ATP. Right? And this you have to incorporate in the system. It's non-trivial, right? Because having this ATP, you have modified enzyme, so it will use ATP and will work much more precisely. right? Or you have to maybe something else to do, but you have to do ATP. If you don't use it, if you use it the way it is, it's no chance to achieve close to 100%, right? All your system and your biology work with error is fraction of percent, right, in your cell, because it's everywhere use ATP. Or it does something a priori not very precisely, but all modification, everything works with DNA, use very high degree of precision. Otherwise, you will not function. This is a major, by the way, point in life. Yeah, you have to have replication process with very low error rate. Otherwise, it's just you cannot make selection, you cannot do it properly. And so this what it seems to me. I mean, yeah, I'm certainly neither a biologist nor a physicist, but very naively, as I understand this situation, it is a major thing. You have to couple this on one hand, and then you have couple it to the active continuous evolution using phases or using something else, and everything might be, of course, controlled by computers. It's kind of obvious machinery you have to create, and then the product will come. But this takes time to do it. It's like the genome project. It took about 5-6 years to implement it, and then and then consequences was coming one after another. And here we are on this trend, and presence of mathematicians there would make it faster. For example, people who search genomes of them, and they found that maybe if you start seriously doing this, not mathematician, kind of abstract mathematician, but practical mathematician, start participating, analyzing all this data which exists there. And there are many things you can do to make it faster. As a theoretician, not mathematician and proving number theoretic theorems, but mathematician as doing something with numbers, with data, etc. But with the knowledge of all that. Without this knowledge, it would have empty kind of absurd activity. You have to learn this biology as much as you can and use mathematician's skill, ability to do that. And this is a kind of most inviting thing for if I was young, I would do that for sure. Yeah, this is the most exciting direction in science, when you can do real discoveries, not just proving theorem, but really discover new phenomena in life, which is certainly fun to do. But this is very recent. I'm saying it all came up to reality in the last 30-40 years. It's never was any chance to this to happen before. And so this is, but this is a kind of structurally it's unclear how to do that if you're a young person, and how to go that. There are obstacles in the system of the educational system, grants, whatever, against it. It's not, and this I spoke to people who actually, unlike myself, participate actively in that, and they complain that it's very difficult to invite young people, because before you start writing papers, you have to 5 years, as a mathematician, 5 years learn biology. And it's unclear actually what happens after that. So it's still a very small field where there are theoreticians effectively working there. But they do that, and some of them who discovered that was Kunin, actually, who was my informatician, who discovered this, not he discovered, but he properly interpreted this discovery with CRISPR. CRISPR, this is palindromic sequences. And he realized they must be related to immune system. And again, you have to be, you see, it's funny, it's kind of unrelated on one hand to something like evolutionary biology, but if you think about that and you realize that, critically think about that, about all these people say about evolutionary biology, you just be critically realize what they say is maybe not quite true, and be ready to see it in the world, and you see it, yeah. So just all the scheme, of course, very naive, and the same when people say that kind of something that doesn't correspond to Darwin, the same is in the nuclear physics. They say it doesn't contradict to Democrat. I mean, it's the same level. Yeah, knowledge of Darwin or biology is modern knowledge. I mean, infinitely far from another. I don't think it was this and close to this. Actually, some mathematician had this mentality and would be not that surprised, like Maupertuis, for example. Yeah, but people I can imagine thinking in these terms. he has this kind of philosophy agreeable with that, but certainly not biologists. Biologists has very poor and kind of feeling for mathematics, as experience with story with Mendel shows. Yeah, you know, when Mendel made this fantastic thing, this numerical relation between some probabilities, and that was not impressed. But nobody was impressed, including Darwin. I mean, they absolutely missed the point. And then, how much 50 years later they couldn't kind of mathematically understand consequences. You know this. Maybe because for your sake I say it. Yeah, that then there was some consequence of this Mendel, and they said that there is no evolution. I'll tell you in a second what it is. And I just want to repeat it. This is a piece of really mathematics. And then actually Hardy just realized what it is and still didn't understand what was happening there. Alec, you listen, I'm saying it to you, yeah? So this mathematical point, the point was that when you look at a kind of evolution, that in certain situation something changes, but on the second round it becomes stable. Some operator in evolution is important. It's square equal to itself. And the operator is as follows, and this is, which again, actually, Hardy did it for 2 by 2 symmetric matrices, but it's true always. If you have a matrix, take some element, take sum here, multiply. You have a new matrix. And normalize, so sum of elements, say 1. Right? So this operator, take a matrix of any shape. In each spot, you take sum, and multiply. So I have a new matrix. So from matrix, you go to a new matrix. This Mendelian operator. And then m squared equals m. And this is not a big deal, it's called just. But actually, Hardy couldn't, no, he wrote a specific formula which was known for 2 by 2 symmetric matrix, right? A. Yeah? With some ABC, something called quadratic formula, and he wrote this formula. He said, oh, multiplication table mathematics, but he didn't realize that just this, of course, special case, actually more general. And this, of course, very simple. I mean, just associativity with tensor product, essentially, or something like that. And this is, biologists couldn't digest it. Of course, they didn't have it in this form. They have it in terms of numbers or gammas, whatever. But they have very poor feeling for mathematics at the time of Darwin and so. But Maupertuis or Buffon certainly had this mathematical perception of things, but that was 100 years before. But in 19th century, I think biologists hardly knew multiplication table. I think so. Exactly, it was a century before, it was different, yeah. There was some kind of, they were not professional people, and they were kind of rather polymaths who worked in biology, and they understood mathematics and physics, but then become specialized and become very blind to mathematics. Now again, mathematics came in a simple form. But this, I think, still, however simple, I think it's quite a beautiful theorem. And it's still, even now, people look at the Wikipedia, I don't know, it's never written there. They still kind of write kind of idiotic formulas. They don't write it in this form. It's incredible, yeah, because it's just repeating. Because no mathematician, I think. On the other hand, there is a development, maybe you make more interesting Jordan algebras, yeah? It's just exactly a generalization of that. This is some special case of Bernstein algebras. And the Bernstein algebras, this is one example, and Jordan algebras is another example. In Jordan algebras, you may love them. And the same kind of mathematics involved. It's very simple, but still kind of rather amusing. There's some formula there, very simple cancellation, but here it's kind of obvious. And this is, again, you see, it's absolutely counterintuitive. It looks like a polynomial map. And polynomial map squared cannot be the same map. Right? It's really kind of almost impossible. But it's not polynomial because you normalize, so it's rational map. And this is negligible. You change a little bit, but then it's possible. and also supersymmetry, or algebra supersymmetry based on the same identity. So it's a rather common thing in a different kind of math. Okay, so we finished for today. Okay, I was a little bit late.